Okay, let's make this nice and big. And yes, we are recording. Fantastic. So let's do an intro to this paper. So this is the Oxford vaccine, or we can call it the chimpanzee adenovirus, and the strain of that is Oxford 1 vaccine, and it has the novel coronavirus 19, which is to say the SARS coronavirus 2 spike protein. So they named it this. They actually came up with the idea for this before the virus was officially named, which is why they're doing it that way. But you can see right down there what it is. So there we go. The idea behind this is that you start out with a virus that is good at infecting chimpanzees and can also infect humans, but is not normally in the human population. And the idea is that if you try to vaccinate with a virus that people are already immune to, they will react against your vaccine and basically destroy it before it can have any effect, which is clearly not what we want. So that's what they're doing here. They are basically hollowing this out and putting a gene inside of it for the SARS coronavirus spike. And they are then launching this thing at cells. I believe they're injecting it into people and allowing the virus coat from the chimpanzee adenovirus to deliver the piece of DNA to cells in order to get people vaccinated. So, what do you need in order to do this? Actually, not very much. So, we're going to be talking a little bit about different symptoms, but I think you can probably work these out for the most part on your own. So what you want to watch for on symptoms, so definitely look at the percentage of people that are showing a particular symptom, but also you want to consider the severity of the symptom, because things like shots do hurt. Normally you get a little bit of pain and a little bit of tenderness at the site of injection. Quite often there will go a little bit of sort of warm feeling around there. These are just the general symptoms of an inflammatory response, and there are actually 5 of them, and that's 3 of the 5, and I bet we hit the other 2 of the 5 somewhere in here. We want to watch out for cases that are either severe or require hospitalization. Those would be red flags, and they've helpfully coded them in red, yes, in case you need to know. And this is a kind of an arbitrary scale. It's whatever the clinicians involved think, is mild or moderate or severe. Or in some cases, I think with this stuff, it is actually, there you go. Patients are recording it themselves in their little electronic diaries, the people who are actually involved in this. And so you can see some of the other symptoms in here, and the symptoms, so the axis down here is showing us how long since vaccination. And so you tend to get, look, the symptom of malaise, which is like a general bad feeling. You feel lousy, like garbage. That tends to peak about, looks like 1 day after the inoculation. So this is when you're actually starting to make some immune response. And then it seems to go down quite a bit. And the other thing to look at here is that they also looked at people who were vaccinated once or twice, I believe. And actually, maybe that's down in the supplementary. Oh, no, here it is. Here we go. So first dose, here's the immune profile. So you got a lot of stuff going on. Nothing hospitalizable, but some stuff and a fairly large percentage. And then on the second dose, look at that, it's much lower. So we're going to go into that and some reasons why that may be. I think to understand this next figure, figure 3, you actually have to go, and this is rather annoying, but you actually have to go and download their supplementary appendix, as what I believe they call it. This is the additional data, and this is where you find out who these patients were, these 172 patients. And what we really want to know is, here they've actually coded it out. I think the paper we're going to do next week, not quite as much. But of these, asymptomatic, fine. Looks like there aren't very many of those. Mild, fine. We got a big cloud here. We know they don't make very strong responses. And then severe, we've got all these blue dots, or blue boxes rather, that tend to be up in the top half. So that's going to be worth looking for. And what we're trying to do is evaluate these, the way that the FDA would evaluate them. And we want to compare them, the 3 vaccines that we're going to cover being the inactivated vaccine, the Oxford adenovirus vaccine, which will be typical of any adenovirus delivered vaccine. And the third one we're going to do will be the Moderna vaccine, and that's going to be in next week. So then what are the terms you're going to need? Over here, prime means a first injection with a vaccine. Prime boost means you give them a first vaccination and then you give them a booster shot later on. And you'll have to look and see what day they boosted. Ooh, there it is, boosted at day 28. And so then, yeah, it looks like we get up to a nice, decent level, which is great. And I believe we are looking here at virus neutralization assays. And so we want higher numbers, which means you can dilute your serum further and actually still get neutralization. They're looking for the point at which, so VNIC 100 is virus neutralization, the inhibitory concentration needed to knock out 100% of the virus. So basically to fully block virus from entering. That's what we're looking at right here. And yeah, and this is a different assay underneath here. Then you've got another neutralization test. They're using pseudovirus, so like a fake virus that has coronavirus spikes on the outside. And you can look and see what kind they're using, but it would be something like the VSV with coronavirus spikes, or some people will use a retrovirus like HIV with coronavirus spikes, and that's fine. Either way, as long as it's the spike protein sticking out, you'll get a fairly reproducible number is the idea. Although I see pretty big differences between the kinds of values they're reporting here, which seem to average around 500, and the kinds of values they're reporting over here, which seem to top out at about 50. Yeah, so that's going to be worth discussing and trying to figure out. Okay, and then the last assay here that we're going to discuss, we're going to sort of stick to the figures for the most part and the interpretation of those. So you've got the T cell response here. And so this is going to be our ELISpot assay. There we go. And we're looking for which cells are starting to make interferon gamma. So this is going to be a variety of T cells. It's like a total reactive T cell count, you can think of it. And you can go have a look. If I recall correctly, and I would like somebody, I would like you to see if you can figure out what the normal level of reactive T cells is that we should expect after COVID-19 infection. So that's going to require just a tiny bit of digging, but there was a paper that came out, at least in preprint form, I want to say about a month ago, that covered this very nicely. And so have a look at that, and that will give you some context how to identify this. Most people report the number of reactive T cells as a percentage. Here they're saying spots per million cells, which include T cells. And so if they were at a 1% level, that would be about 10,000 per million, just for reference. So there we go. That is the end of the big part of this. And yeah, remember, for a couple of these things, and particularly any methods questions behind here, you're going to need to have that supplementary appendix, I believe they call it, the online supplementary information, this appendix page 3 right there. That's what you're going to need, and this is what we're going to talk about. So thanks very much.